Hi to all of you who are seeing this video. So we'll be continuing with the molecular biology lecture series. This is the ninth lecture of the series, and in this we'll be discussing the replication process, its three phases: initiation, elongation, and termination, in particularly E. coli taking as the model. So before we begin, if you have come to this lecture directly, you should know about the processes, the reactions taking place in the replication, enzymes involved, what is the difference between a replosome and primosome, what are helicases and their function, and different DNA polymerases in eukaryotes and prokaryotes, and what does the clamp do in prokaryotes and eukaryotes. If you don't know this and you have come up in this lecture directly, so you have to go through the previous lectures as well to know all about it. The process of replication in itself in both eukaryotes and prokaryotes comprises of three stages or phases: initiation, elongation, and termination. We'll be discussing all three of them in this lecture. Replication process, the DNA fragment which undergoes the replication in itself is called as the replicon. So this is the fragment of a DNA or a whole genome which undergoes the process of duplicating itself into a daughter ones. So replication of DNA in a replicon is catalyzed by the polymerases at a particular site which is called as the origin of replication. The origin increases the efficiency of initiation of all the enzymes at a particular site, and they require in turn the protein factors and the enzymes before the polymerases in itself can duplicate the DNA. Origins also provide a target for the regulation of the replication and is the only phase in replication where the regulation also takes place. So in a replication, before the actual process of replication, the factors, the initiation factors, and all other enzymes like helicases, topoisomerases, primerases has to bind and identify the DNA fragment where the replication has to start. This is the initiation or the origin of the replication site at which this happens, and the whole process is called as pre-replication process. So in a process of replication, these are the enzymes or the accessory factors in addition to the polymerases which are needed. The model here, the table here is given for the E. coli initiation phase of the replication. Different proteins are listed here. Particularly what we need in the E. coli cell is the DNA A protein, DNA B protein, and DNA C protein. Those are the helicases ones, and the single strand binding proteins are needed in addition to primases and HU like proteins. Then DNA gyrases, the RNA polymerase in itself, and methylase all play a role in the pre-replication process so as to start the replication at a particular site. Those are called as origin of replications. In E. coli, the replication takes place in a manner as is seen in the figure, that it takes place at a single site which is called as ORI origin of replication, and it moves bidirectionally, one side here, one side there, so as to peel like it feels like a peeling of the daughter DNA on the parental DNA. And once you terminate the replication or complete the replication, and what happens is that the two copies are produced, but they are intercalated into each other, which require topoisomerases to separate them to form two molecules of a DNA in the E. coli cell. In prokaryotes, as happens in E. coli, the replication origin is designated as ORIC, origin of replication, numbered as C, and it usually consists of the highly conserved 245 base pair DNA sequence. And within this 245 base pair sequence, there is a repetition of certain tandem repeats. Those are called as iterons, as you can see in the figure. The blue ones are the 9-mers or nonomers. They are present as 4 copies, and they have a sequence of TTA, TCC, ACA. And then the yellow ones are the 13-mers conserved sequence iteron, which are present in this 245 base pair ORIC region. as 3 copies. So the sequence of this 13-mer is GATC, TNT, TNT, triple T. These are the highly conserved in the prokaryotic regions, and they are the ones which are melted first during the replication process, so as to start the pre-assembly of the whole replosome factory to begin the replication process. So the initiation of the replication happens in 3 steps. By the identification of the replication machinery, these nonomers and these 13mers have to be first identified. And at least 9 different proteins play a role in the initiation phase of the replication in prokaryotes. The 9-mer sequence is the site at which the first proteins bind. Those are DNA A initiator proteins, as you can see in the accompanying diagram. The DNA A protein, together with the energy from ATP hydrolysis, identifies these nonomers and binds to them and loops it around themselves. Usually 20 to 30 DNA proteins bind to this 4 nonomer sequence and wounds it over himself, and then in second phase the histone-like proteins together with the ATP again binds to this area and melts the AT-rich regions so as to form a separate single-stranded molecules out of the melting of the bonds between the 2 strands. Then what follows is the on each strand 2 hexamers of DNA B and DNA C complex binds on each of the strand here, as you can see in the picture, so as to stabilize the unwound form of the strands. Then SSBPs bind to each strand, preventing the rewinding of the 2 strands, and stabilizes the 2 strands for the synthesis of a DNA on them by the help of a polymerase. In prokaryotes, the polymerase is Pol3. In eukaryotes, the polymerase will be Pol alpha first. So the before the initiation takes place, these 3 steps has to be followed. So this is a sort of a pre-initiation to the polymerization in replication. And in eukaryotes, usually DNA A, DNA B, and C take place, take the involvement in this melting of the initial site AT-rich region of the 9-mer sequence within the 245 base pair conserved sequence in the origin of replication, ORIC. One of the fascinating factors of the replication initiation is that it is the only phase in which the regulation takes place. The regulation is actually met out by the DAM methylase enzyme, the DNA adenosine methylases, which methylates the N6 position of the GATC palindrome. So the adenosines present in the GATC have to get methylated by DAM methylase so as the application takes place. So only those DNA strands will get replicated which are methylated completely in both strands in the adenosines of the GATC palindromes. If however there is a hemimethylation of the origins, that is the some of the or many of the adenosines in the GATC sequence are not methylated, or only one strand is methylated and another is not, this is called as hemimethylated origins. For these kinds of DNAs, the replication cannot take place. In eukaryotes, as an example, in yeast, the origins are called as autonomously replicating sequences or replicators. They are usually 150 base pairs in length and contain several conserved sequences. The minimum length of conserved sequences which supports the replication initiation is found to be 11 base pairs, as is shown in the slide. Also in the diagram on the right hand side, the sequence is AT-rich region, as you can read yourself, and they are located all over the length of a genome, spaced at 30,000 to 300,000 base pair apart. So there are multiple of ARSs in the eukaryotic genome, so that the initiation of a replication take place concomitantly at all sites in one go. For the initiation process, similar to that of the prokaryotes, we need origin recognition complexes. They are usually the multi-subunit proteins which form the assembly at the ARS after recognizing it. That subassembly is called as pre-replicative complex. And usually this subassembly takes place at the beginning of the late M phase or early G1 phase. ORCs are usually hexamers of related proteins. They are ORC1, 2, 3, 4, and 5, and 6. When ORC binds to the ARSs after recognizing them, it recruits the two cyclin-dependent kinases called as CDC and CDT. After binding with the cyclins, the ORCs also recruit six more proteins that are called as MCM 2, 3, 4, 5, 6, and 7, which bind to it and form a whole assembly of 14 proteins, which are called as mini chromosome maintenance function complex. And together, all these make pre-RC, pre-replicative complex. Pre-replicative complex is then activated to do its function of initiation of replication by cyclin-dependent kinases. Cyclin-dependent kinases, what do? They actually ask and recruit the MCM10 and then cause the phosphorylation of the MCM, which are already present there, MCM2 to 7, any of the 5 are phosphorylated. As soon as the phosphorylation of these mini chromosome maintenance function complex happens, the pre-replication becomes a replication initiation, and then the replication moves further on to initiation process. The elongation phase soon follows the initiation phase of the replication and is simply the extension of the daughter DNA on the template strand. Based on the information and the complementary rules, the polymerases synthesize the daughter DNA in a 5' 3' direction. The 3' end of the daughter DNA is the end which is extended. There is another difference between the two strands, is that the lagging and the leading strand. Lagging strand usually is synthesized in a discontinuous manner in retrospect to the leading strand. Leading strand is synthesized in a continuous manner. In a lagging strand, there is a discontinuity, and the fragments which are formed at every interval are usually 100 to 2000 base pairs in length. Those are called as Okazaki fragments, which are joined by DNA ligases. Furthermore, we know that the lagging and leading strand synthesis uh, is usually looped around the DNA polymerase by 180 degree. That's why it usually lags the leading strand. I have already cleared it in the previous lectures. So the elongation phase also we know that requires the RNA primer to synthesize the lagging strand every time, and this is done by the primases in the prokaryotes, while as it is done by DNA pol alpha in the eukaryotes at the lagging strand. If you don't know this information already, you need to go through the previous lectures as well. This slide I have already covered in the previous lectures, but to quickly go through it, that we know that in prokaryotes the primase action is by DNA G protein, and after the primer is synthesized, the extension is done by the DNA polymerase 3, which is the main polymerase in the prokaryotes, and it also has a 3' 5' exonuclease proofreading activity. While as in eukaryotes, we know that DNA polymerase alpha is the first one which begins the primer formation because of its integral primase activity. Then it extends it to some 14 nucleotide of DNA, which then is taken over by DNA polymerase delta by polymerase switching with the help of a replication factor C. And we know that the difference between the leading and lagging strand is because of the discontinuity. For the lagging strand, we need a primer only once synthesized, while as for the lagging strand, we need the primer to be resynthesized again and again and again at an interval of usually 2000 nucleotides spacer. And this activity is done by primosome, which contains the primases as well as helicases, and they are always associated with lagging strand till the end of the replication. The third and the final phase of the replication is the termination. It is the phase where the replication forks are arrested at a particular sequences which are called as the terminus sequences. They are 20 base pairs sequences which are highly conserved, and they are present as multiple copies on both sides of the circular DNA in the E. coli, usually present around about 180 degree to the origin of the replication in the E. coli cells. What happens is in these TR sequences, the protein specific protein binds to them, which are called as TUS proteins, terminus utilization substances. They are the proteins which arrest the replication for coming on either side of these sequences. So which one of them will come first will be arrested first and will bring out the termination. And finally, the termination is brought about by with the help of an topoisomerases. Topoisomerases, you know that they are the one who relax the torsional strain, and they are the ones also needed here to decatenate the two copies of the DNA which have been just synthesized by the replication machinery. The two copies of the DNA are interlooped into each other. These are called as catenates. And for the release of them as a separate copies, we need topoisomerase function. Here, the topoisomerase 4 is involved in the decatenation process because it can change the linking number by plus minus 2. Usually in DNA gyrase functions in prokaryotes, and topoisomerase type 2, usually 4, named as 4, but it is a type 2 topoisomerase which decatenates the two copies of the DNA and hence brings out the termination in essence by releasing the two separate daughter DNA molecules. So this was the essential story about the replication process. In the next lecture, I will be discussing the transcription. If you like the contents of the video, you can order the book Molecular Biology and Biotechniques from Amazon. It's available both in Kindle and paperback format. And do subscribe to my channel for further videos which I will upload in due time.